My name is Trey Ideker. I'm a professor of medicine, computer science, and bioengineering at University of California, San Diego. It's my great pleasure today to introduce the Cell Maps for AI project, a functional genomics data generation project, which is part of the NIH Bridge2AI program. As I think many of us are already aware, AI and machine learning have made tremendous strides over the past few years in revolutionizing many tasks in society, including those in biology and medicine. And in particular, today in a functional genomics project, we are most interested in questions that relate to interpretation of the human genome. Questions such as, how does the genetic code dictate the architecture of living systems? How do genetic changes to this code and thus that cell architecture promote or prevent risk for disease? With artificial intelligence and machine learning systems, one often thinks of input-output relationships. And so here, quite simply, you might, as the input to such a system, include the genome sequence itself, as well as relevant patient and clinical history and other information about the individual as appropriate. You would then translate that using a first-stage machine learning system to impacts on the molecular, structural, and functional architecture of cells and tissues, and then changes in that human architecture would be translated to predicted behaviors, risks for disease, treatment responses, or other phenotypes of interest. Incarnating such a system practically might amount to talking to your iPhone or your GPT algorithm, where you would say, dear AI, my patient has the following mutations in these genes. This is the case of a cancer patient, and it's not simply one gene mutation. Typically, it's dozens to hundreds of mutations and variants one is grappling with there in those genomes. The questions you might want to ask relate to what treatment is best, or if you're in a pharmaceutical company, should I be looking at any new targets? There's already quite a lot of data that have been placed in the public domain relevant to training artificial intelligence systems for these types of genome answering systems. In particular, over the past 20 years since the first human genome sequence, we've now seen deposited in the public domain, or at least in domains where the data are accessible to researchers, millions of genome sequences of human individuals linked to thousands of human traits, including risk of disease and treatment responses. So while many more sequencing projects are continuously ongoing, and those are important contributions, we felt that a particular lack or gap in data that could be used to train AI systems in genome translation relates to the molecular level. How do these genetic changes at the left translate into changes in the structure and function of molecular systems in cells and tissues? There certainly are data available in some degree here, but we felt this was an area where much more data are needed, and that's the focus of the Bridge2AI Cell Maps for AI program. This is akin to thinking about the genome as revealing 20,000 genes, but those genes are simply parts of a much greater machine, much like the parts of this Golf here are shown splayed out on the factory floor of the Volkswagen plant. What we're after here is for this middle layer of information for training the AIs relates to how those parts are assembled to create cells and tissues, much like the shop manual shown down here at lower left for how the parts of the Volkswagen Golf are assembled. The particular data sets that we are generating to fill in this cell biology gap are shown over here at left, and I'll say much more about them in just a few moments. But they're briefly three types of data. Those major data streams are generated in parallel for large sets of proteins to survey different aspects of human cell biology. The three data streams are integrated together into common cell maps, as shown here in the middle of my screen. And then as these cell maps are developed, they will be used as the basis for building machine learning systems for translation of the human genome. The model cell types we're going to start with initially and have begun generating data with already are two types. Breast cancer cells on the one hand, both untreated and treated with 2 chemotherapies, and on the other, what are called human-induced pluripotent stem cells. These are models of the embryonic cell state, which of course can develop into all human cell types and tissues. And here too, we'll look at the unperturbed state as well as neuron differentiated iPSCs and cardiac differentiated induced pluripotent stem cells. Both these cell populations have been ethically sourced with attention to human diversity. They will be interrogated by, as I mentioned before, three main experimental platforms to measure the organization of human cell biology. First is confocal protein microscopy to localize each protein in the cell population and subcellular structures. The second is a biochemical complementary approach based on affinity purification mass spectrometry to localize proteins relative to others in human cells. And the third is a functional approach based on the CRISPR-Cas9 system to perturb each of those proteins in turn and measure the effects of those perturbations on cellular state. Here's an example of those three data streams. For one protein in particular, the protein known as SMARCA4. SMARCA4 is an important factor in the control of transcription. It's a transcriptional regulatory factor. Over at the left, you can see its localization in human cells. These are the breast cancer cell population I mentioned. In the middle here, you can see the interacting proteins of SMARCA4 is determined by affinity purification mass spectrometry, which themselves, as you see here, form a web of interactions among themselves. And over here at right shows a functional network linking SMARCA4 to other gene perturbations that produce the same cell state. For instance, a perturbation to BRD7 looks very similar to a gene knockout of SMARCA4 in terms of its effect on the cell overall. But that's, of course, just one protein. And we here in the CM4AI are rolling through these technologies over large numbers of proteins to survey human cell biology. Here is an example of what such a map is starting to look like. This is a hierarchy of protein subcellular components or cellular components we can identify or resolve in those integrated data streams. Overall, we can see 275 discrete assemblies of proteins, which factor into a resolution of organelles at the top level, and then within each of those organelles, smaller and smaller protein systems represented by each of these circles as we move down in the hierarchy. How might such hierarchies of systems that we can resolve be useful for machine learning systems to interpret the human genome? Well, here is one example or a proof of concept of how this is going to work. Here is an example protein assembly known as collagen. As you might be aware, collagen is the primary constituent of the extracellular space that binds cells together. Collagen forms or is formed by the coming together of groups of proteins or collagen subunits, typically in threes, to form this triple alpha helix, as shown up here at the upper left. It was not previously known that collagen is under mutational selection in cancer, but it turns out when you take this protein assemblies view or a subcellular components view to interpret the cancer genome, what you see immediately is that 70% of melanomas and about half of lung adenocarcinomas, among several other tumor types, mutate collagen complexes quite frequently. The reason this had never been noticed before this paper down here at the lower right is because these mutations are spread out over all of the possible subunits of this collagen machine, such that at the gene level or at the level of each one of those subunits, the mutation is relatively rare. So that will hopefully give you an overview of the premise of our study and some of the progress that's ongoing. A final note on the leadership of this project. We are a consortium of more than 10 institutions in the United States and in Canada. Our leaders are focused on 4 top-level goals. Goal 1 is to generate multimodal data revealing the architecture of human cells. Goal 2 is to build the corresponding tools that enable AI-ready cell maps from these integrated data streams. Goal 3 is to engage and educate a broad community of stakeholders in these maps, and as a corollary, to think deeply about the ethical issues that are associated with translation of the human genome and how bringing forth knowledge of human cell biology in artificially intelligent systems can help. And finally, the last goal is to widely distribute the data maps and tools with attention to diverse audiences of individuals and ensuring everyone has access to benefit. So with that, I will close and call your attention to the other seminars and recordings that you may find as part of this collection. Thank you very much.